So our last speaker in this session is Professor Michael O'Sullivan. Mike has joined our sunny shores in Australia a couple of years ago, and he's a neurologist and clinician scientist, and he's currently Professor of Clinical Neuroscience at the University of Queensland. His main interest is in networks, brain networks that support cognition and injury to these networks, with a big interest in memory and models of memory. The aim of his research is to understand impairment after injury and mechanisms of resilience and restoration of function. And today he's going to talk to us about white matter microstructure and neurodegeneration in the memory system. Chicken or egg? Tell us.
Thank you, Amy. Thanks very much. And thanks very much for inviting me and asking me to come. And you've given me a tremendously difficult job to follow that tour of very impressive, rich data sets, so beautifully analysed. But I will try to do that. And also, thank you for asking me to talk in the vascular risk part of the day, because it's a real opportunity to show you some new data about the memory system and the potential effects of vascular risk. So at various points in the session this morning, we've focused either on the cortex or the grey matter of the brain, or on changes in white matter structure. But in very many of the situations that we're describing, changes in those compartments go hand in hand. And in some circumstances, we feel very comfortable about what the direction of effect might be in the relationship between grey and white matter change. So examples of that are if we see a person who has a white matter infarct, a lacunar infarct, and we see thinning of the cortex in the regions likely to be connected with that infarct, we feel quite comfortable that the direction of effect is white to grey, and that this is a dying back of axons that has some effect on the cellular structure of the grey matter. And equally in established Alzheimer's disease, people have generally taken the conclusion that the disease is predominantly damaging neuronal cell bodies in grey matter, and that the white matter changes are somehow secondary. But there are a number of situations where that direction of effect is less clear, and we're less confident about exactly where the process is starting. And some examples of that are the very earliest stages of Alzheimer's disease, where there's increasing interest in very early white matter change, and also in people who have disease-free aging, where we see changes in the structure of the brain, but a lot less clear on the mechanisms of that. So I'm really going to talk about some new data and look into this in a bit more detail, particularly in relation to the memory network in the brain. So I'm just going to introduce the memory network first, and then talk about some data about how those changes interrelate in the memory network. So when I talk about the memory network, what I'm really talking about are this extended hippocampal network in the brain, an idea that has just arisen from what's more traditionally called the circuit of Pepys. So what I mean by that are the network that includes the hippocampus and other grey matter structures, particularly the anterior thalamic nucleus and the mammillary bodies, and also the connections between those structures, which include the white matter tract called the Fornix, which connects the hippocampus with more anterior structures, then also connections around the cingulum, and at the completion of the circuit of Pepys by parahippocampal white matter connections here. And this is just the Fornix in a little bit more detail here on the right. And this is a structure that connects the hippocampal formation, has direct connections with the anterior thalamic nucleus, connects to the mammillary bodies, and also connects to the basal forebrain and the frontal lobe. And a number of years ago now, we attempted to reconstruct some of those pathways using diffusion imaging. And that's really the background of the data that I'm going to show. And these are just the final results of applying diffusion imaging and tractography to try and reconstruct those connections. And you can see here this tract in red is a reconstruction of the Fornix. The tract that you see in green is what we call the parahippocampal cingulum. So this is this completion of the circuit of Pepys going from the parietal lobe back in to the parahippocampal white matter. And the tract that's in blue is another temporal association tract, the uncinate fasciculus, which hooks forward from the temporal lobe into the frontal lobe. So a few years ago now, we looked at reconstructing these tracts and looked at how they changed in aging, and also in people with early memory problems, to look at how to look at whether structural variations in white matter connections were contributing to cognitive change with aging. And these are the findings really in brief. And what we found is that the Fornix shows clear age-related variation in microstructure. So from the age of about 50, there is a linear change in microstructural measures in the Fornix. And also those changes in the Fornix are the dominant correlate of age-related change in memory performance. So they explain much of the change in verbal recall performance with age, and they also explain some of the inter-individual differences between people of similar age, in that we still see relationships when we co-vary the effect of age out. And if you look at people with early memory problems, people with a diagnosis of mild cognitive impairment, the Fornix is compromised at an early stage, and it's really relatively easy to show change in that structure. So from an early stage, there is obvious compromise of Fornix microstructure in people with mild cognitive impairment. So in terms of the results with aging, the Fornix is the leading player in age-related cognitive decline. What we couldn't really say at this stage was what the origin of those changes in Fornix microstructure were. And this was the question that I was asked every time I presented this. What is the cause of those changes? Are there changes in the hippocampus? And we didn't really have the imaging quality at that point to try and understand what the origin of those changes were. So what I'm going to do now is just talk about 3 much more recent studies that shed some light on the relationship between grey and white matter change in this particular network. So the first of those was done by a PhD student that joined me in King's, called Naomi Hartop. And what Naomi did was just do a simple, straightforward replication of the results from the normal aging study, but using better methodology. So we just recruited a new sample of healthy older people. We imaged them in London. This was a few years later. We had better quality T1 weighted data, so we were able to broaden out the analysis of this data a little bit. The key difference was that we had better quality structural data, particularly better quality T1 weighted data. And because of that, we were able to apply some new methods to look at hippocampal subfield segmentation to try and relate changes in Fornix structure to subfields of the hippocampus. So this is the method that you can see here on the left. So this is an automated algorithm that uses the contours and shape of the hippocampus, which has been developed in a sample of standard brains and validated against anatomical landmarks. And what you get are that you get this parcellation, this labeling of the hippocampal subfields, and you also get measures of grey matter volume within them. So what Naomi found is, first of all, she replicated the association between Fornix microstructure and age and Fornix microstructure and memory. And then she went on to show that Fornix microstructure is strongly related to the volume of hippocampal subfields. And most interestingly, if you co-vary out the collective effects of all of the subfields, you still find a significant relationship with a subfield called the subiculum. And this at the time was very interesting because the subiculum, in one of its roles, is really to be effectively the departure lounge for information from the hippocampus, and it contributes very heavily to connections in the Fornix. So at this point, this fitted a reasonably obvious hypothesis that what this was really all about was loss of neurons in the hippocampus, sending projections to the Fornix, and that's what the basis of age-related change was. Now, the other thing that we could do in this analysis is we can say, well, are these relationships between grey and white matter structure, are they actually important? Are they important for cognition? And because we saw relationships between white matter and all of the different subfields, the question that we asked was, are there particular relationships that are important for cognitive function? And you can address this with regression, because if you regress something like Fornix anisotropy against subiculum volume, that really has the effect of keeping subiculum volume constant, and the residual that you get from the regression is a measure of changes in the fornix in the virtual situation where the subiculum wasn't changing. And if we did this analysis, and we did it in a way that kept the volume of the CA1 subfield of the hippocampus constant, we found that there was still a significant relationship between fornix microstructure and memory. And the converse of that, if we attempted to hold subiculum volume constant, we didn't see a significant relationship. So the conclusion from the contrast between those two models is that the connectivity between subiculum and fornix is important for cognitive function. So at that point, when we did that study, we thought, this is all holding together quite nicely. And what we're seeing is loss of neurons within the hippocampus that are important in normal disease-free aging. But what we hadn't done at this point was really done any analysis of the direction of effect between white matter and grey matter measures. And so this is the second study that I'm going to describe to you, which was a follow-up study done by my colleague Claudia Metzler-Badley in the Cardiff group. And what Claudia did is that they also planned to do a larger study looking at the relationship between fornix microstructure, aging, and memory. But what they also did in this study was collected a substantial extra amount of information, both from imaging and also on vascular risk status. So what they did in this study was, again, looked at diffusion measures, looked at other imaging metrics like quantitative magnetization transfer and relaxometry, and also collected information extensively on vascular risk, including measuring things like waist to hip ratio, body mass index, and measures of central obesity. And this is their imaging pipeline for the study. So they collected information on diffusion imaging, on QMT, and I won't really talk in detail about what those measures mean. They did tractography of the same tracts that I showed you at the beginning, and they also did free surfer-based segmentations of the hippocampus. So the first thing to say from Claudia's study is, again, they reproduced a number of relationships between age and microstructure of the hippocampus across a broad range of measures derived from diffusion imaging. And then what they did was they went on to try and model the relationships between microstructure of the fornix and changes in grey matter structure in the same circuit. And they tried this by using two types of regression model. A regression model where you look at the hippocampus as a mediator of the relationship between age and fornix, and a converse model where you look at the fornix as the mediator of a relationship between age and hippocampus. And what they found was exactly the opposite of what we might have predicted from our study. They found that the models where the fornix mediated the relationship were almost all positive and led to complete mediation of the effect, suggesting that fornix microstructure was influencing hippocampal volume rather than the other way around. The models of hippocampus mediating the relationship with fornix were largely, with a single exception, negative. So this is the opposite of what we'd expected, and this suggests that it's actually the fornix change which is driving the change in hippocampal grey matter volume. Now, the other thing that they found was that in a larger sample, they were able to get a lot more detail about the associations with vascular risk. And if you look at the associations between fornix structure and vascular risk, there are a number of novel things that hadn't previously been described before. And the particular one that I'll pick out is that on several measures of Fornix microstructure, there are associations with measures of body habitus and obesity. So there's an association between quantitative MT imaging and waist to hip ratio, and also diffusion measurements of Fornix structure and waist to hip ratio, which you can see in the top three rows of the graph. Now this actually, we in our original study, we just by accident found that there was an association between Fornix FA and body mass index. It was a small sample and we didn't quite believe it, and we published it in a fairly cautious, interpreting it cautiously kind of way. But this is a much larger sample and gives a much more convincing demonstration that these changes in body habitus really are strongly related to Fornix microstructure. So the picture that emerges from that is the idea that vascular risk states are influencing white matter structure, and that's the thing that's driving neurodegeneration in these memory circuits, which is not entirely as we had predicted from the start. So again, you might think, well, you know, it's just one study and it's a cross-sectional study. There's no true longitudinal data to really back up these ideas of what drives what. So the next question was, is there a way that we can try and replicate this in truly longitudinal data to try and see if we get the similar result? So the way that we did this was that I had a PhD student called Tavia Evans, who was doing some work with the Rotterdam study investigators, trying to replicate some of our earlier results about the functional anatomy of memory. So what we did was we thought we can look at this in Rotterdam scan study data. So for those that don't know, the Rotterdam study is an epidemiological study based in a single suburb of Rotterdam. It's been running since 1990, and to date, it's enrolled just under 15,000 individuals aged 45 years or older. An MRI was introduced into the Rotterdam study in 2005 with planned repeated scans at 4 yearly intervals. And at the point that Tavia looked to do this study, diffusion and structural MRI data was available in about 4,500 individuals from the Rotterdam study. And the Rotterdam study protocol also includes a measure of memory, which is a simple 16-word list learning test. So the advantage of this was that this is a truly epidemiological sample, and this has truly longitudinal data. So we're able to look at MRI at time point 1 and look at what the influences of structural changes at time point 1 are on structure of the brain 4 years later. And this is what we found. So we had, in the final analysis, about 4,000 people with baseline imaging data, about 2,400 of whom had MRI 4 years later. And the first thing that we looked at was we included a measurement of the Fornix into the Rotterdam analysis protocol, and we looked to see whether Fornix microstructure predicted memory performance at follow-up. And we did this using cross-lagged regression models that take account of the interval of time between scan 1 and time point 2. And what we find in that analysis is that Fornix diffusivity 4 years before assessment does indeed strongly predict memory performance. The crossed association between memory and Fornix diffusivity is not significant. So then we thought we can also test our relationship between grey and white matter structure in this longitudinal data. So we ran that analysis, and in this analysis we looked at subiculum volume and Fornix diffusivity at those 2 time points. And what we find in the cross-lagged regression model is that Fornix diffusivity predicted subiculum volume 4 years later, but the converse association was not significant. So again, in truly longitudinal data, in healthy, disease-free older people, these models suggest that changes in white matter structure are driving changes in grey matter in the same memory network. So to summarize all of that, changes in grey matter and white matter structure are intimately related in healthy older people. We showed in Naomi's study that the relationships between grey and white matter structure are important in determining cognition in those people. And to date, at least, the evidence from the statistical models is most supportive of a white matter to grey matter direction of effect. And we've seen that both in a cross-sectional single-center study and also in the longitudinal data from the Rotterdam study. And finally, we see that Fornix microstructure is associated with vascular risk factors, particularly those that are related to body mass index. So the question is, are we seeing degradation in core memory networks that is actually being driven by vascular risk, which is having an effect on white matter connections in those networks and then driving degeneration, which is certainly against what might have been suggested a couple of years ago as the most likely model of change that we're seeing. But we do have to remember a few caveats associated with that. The idea of causal inference from regression models is challenging. There are issues about hidden confounding variables and whether you may have actually identified the true mediator or whether the mediation effect is arising from hidden confounding. But the second major caveat is that we don't really have anything to go on in terms of what's going on pathologically. We don't know exactly what's happening in the subiculum in aging. The pathological data is equivocal. There are few high-quality studies of subiculum cellular changes in aging, and we just don't really know exactly what the underlying pathology might be. And if we knew that, then we might have some more confidence about what the actual mechanisms driving these associations are. And I think that this is one area where translation from model systems in animals is very important, because the thing about model systems are that you can intervene and interfere with some of these variables, which take away some of the challenges in terms of causal inference from those changes. So I think, these are interesting, they're overall provocative results, but I think there's a lot of work to do to really understand what's happening at a mechanistic level here. So I'll stop there, and I've thanked the various people who did the work on the way through. So I just want to thank the people who funded the work, the MRC in the UK, and also the CoStream grant is a European Union Horizon 2020 grant, which may end on the 29th of March, at least for me, but who knows. Thank you very much.
Really appreciate, Mike, that talk, because I think that in his presentation he's just beautifully outlined some of the promise and the opportunity, but also some of the limitations of imaging, that, tension between covariance and causality, and also how much the model affects what we actually find, which is sometimes a bit of a bugbear in imaging research.